שלום לכולם. טוב, כל הכבוד למי ששרד לשיעור האחרון. טוב, לפני שנתחיל, מישהו רוצה לשאול משהו, להגיד משהו? אוקיי, אני הכנתי שתי מצגות להיום, ויש לנו הרצאה של לנה ובר. אז אני חושב שנתחיל ממני, ואז נעשה כזה כדי שלא יהיה משעמם. נתחיל ממני, נעשה הפסקה, ואז נחזור אליי, בסדר? אלא אם כן אתם צריכות ללכת או זה, או שאתם רוצות כאילו, לנה ובר, אתם כאילו יש משהו, אתם רוצות להתחיל, או שזה בסדר מבחינתכם?
כי אני אשמח לעשות את זה כמה שיותר, כאילו, מוקדם במהלך השיעור.
אוקיי, מה, כי אתם בלחץ ואתם רוצות לגמור עם זה כבר?
כן.
אוקיי, אתם רוצות לתת משהו. סבבה, אז אתם תתחילו. אבל יש לנו הרצאה, אז אנחנו נתחיל בהרצאת אורח, ואז תשמעו אותי ברצף. מצטער.
אני רק אעלה את המצגת. Okay. Can you see my screen?
Yes.
Okay, I'll just go. No. Okay. So, hello everyone. Our names are Bar and Lena, and we would like to present today the KCML framework. It's a machine learning framework for inference of multi-scale gene functions from genetic perturbation screens. This paper is pretty new as it's from 2020. So what is KCML? KCML is a machine learning framework that maps the connections between genes, functions, and phenotypes. And we'll talk about these connections more in depth later on. It can be generalized to different types of data and predict novel gene functions based on the variety of data. So a quick explanation on what ontology is, since we'll be using the term later on. Gene ontology describes our knowledge of the biological domain. Gene ontology is also the name of the world's largest source for information on gene functions. We can see here an example of genes in the database, like URB1. Go terms or classes are biological processes which the different genes belong to, and they're context independent.
Genetic screening is very useful for characterizing gene functions. It is based on the gene knockdown principle. Each time we knock down a certain gene and see the phenotypic behavior on the screens. So, for example, this picture shows us wild-type cells stained with the DAPI to point out the nucleus and the green for the cell area. If we knock down gene 1, we receive the image on the right, which depicts how the unactivation of this gene affects the cell. So each time we knock down a different gene and see the influence on the images on the right. So here we see the gene 2 and gene 4, they have similar phenotypes. So we can understand that they are connected. And two more ways that we can learn, except for image-based manner, we can learn about gene functions is the viability and the gene expression. And in the paper, they use them to validate the results. So gene perturbation datasets contain phenotypical data for each gene. The features are a visual description of the result of losing a gene, These datasets are usually analyzed with clustering methods to identify quantitatively similar phenotypes. So we try to identify the function that each cluster represents. But in fact, this approach is pretty difficult and doesn't work well in practice. So in many studies, we see that most data is not analyzed. As for today, the function of a large number of genes is still unknown. And even the ones that we do know can change after perturbation. So there's a need in a context-driven tool to discover new gene functions, which are important to understanding biological processes. So now I'll present the main idea, which is the novelty in this paper. We have the gene after perturbation in its new context, and we would like to find its new function within this context, the new tissue. So first, we take the cells which express this gene from that tissue. We stain them and extract the phenotype. Then we search for similar phenotype, which is the signature phenotype of a known gene. And this information is found in gene ontology notations. And this is done by classification method. And then we apply the new function on the original gene and hypothesize that this would be its new function in the new context. The area inside the green mark is what the KCML algorithm actually does. So now it's the explanation about the KCML algorithm. First, we have the input. The input is the data set of what we know about genes involved in a certain process. It's a term. Let's take cell cycle, for example. So these data sets contain a positive class and negative class. Positive is for gene known to be involved in a cell cycle. And the negative class is a random set of genes. And the classifier on the next step tries to find if these genes have different signature phenotype from a random set of genes. So in this paper, they tested random forest binary SVM and another method. And they decided to use the SVM because it resulted with less overfitting. But the metric data is not mentioned, so we didn't like it. But anyway, the SVM identifies discriminating features through a separation plane. And after training the classifier, we apply it on a new set of genes and try to predict their functions based on the phenotypic similarity. So we do it for different terms, different biological functions, like hormone activity and apoptosis. So some phenotypes are hard or impossible to capture, so some classifiers, they will not find a signature phenotype, and they will not be counted in the final prediction. So eventually, we aggregate the results of all the classifiers, and we receive the output, which is the multiple predictions on different terms or different gene functions of the set of genes. I hope you understood. Here we see the data used in this paper. It shows us the generalizability of this algorithm. The one is the gene expression. Second one is the image-based. And the third one is the dataset of the viability. I think I missed a slide, sorry. It's the analysis pipeline. First, we use large-scale genetic perturbation screens. Then we use it for the...
הטלפון שלי התחיל, מה זה?
אני לא שואל אותך. אני החלפתי את עידית בגלל שהיא הולכת לוועדת חוץ וביטחון לבחירת יושב ראש, ואני מודיע לכם שכל עוד אני מנהל.
סגרתי את הכל ועדיין הטלפון המשיך לעבוד, התחיל לבד, אני לא מבין למה. קיצור.
לתת לי לשתות מים.
מה?
כדי שאני אשתה מים.
מצוין, אני מצטער. טוב שפדיחות כאלה קורות בשיעור האחרון, שאני כבר לא, שום דבר לא מלחיץ אותי.
So the analysis pipeline, we take large-scale genetic perturbation screens. We apply the KCML algorithm that classifies the gene perturbation profiles, and then the classifier outputs predictions of gene functions. And then we can make a data-driven functional map, which lets us map connections between genes based on their functions.
All right. So a few things that we can see about the KCML algorithm. The first is that it outperforms existing methods for a classification, such as k-means and self-organized maps. This, as we can show, the other methods randomly classify the functions, while KCML identifies functionality relevant phenotypes, as existing methods don't provide a clear interpretation of the quantitative phenotypes. Something nice that they did in this paper is how they validated the process of the framework. They first downloaded uniquote annotations up until March 2018 as their training set. Then they downloaded annotations from March 2018 until July 2019 as the validation set. And they were actually able to predict many of the annotations from the validation set. And they showed that actually the algorithm is not biased to a specific data sets. And they do have a high percentage of validations. KCML can actually also classify biological functions that act at different scales from the molecular to the tissue level. The authors were able to create a network representation of classifiable terms, showing the connections between them. We can see it in this graph here. A nice connection that they found using KCML is the connection between olfactory receptors, which are smelling genes in the gut, and colorectal cancer. And they suggested using these receptors as biomarkers to find the cancer. So to summarize everything up, KCML allows a systematic analyzation of large-scale genetic screens. It selects biologically relevant features, and it can identify phenotypes and link phenotypes to functions. As time advances, it can help us understand gene functions at the different types of levels and the connections between them. The algorithm is flexible and generalized to different types of data sets, as we previously said. However, this research does perform on a small set of data in a very specific domain, and further research is required. We also suggest using different features which can affect the phenotypes, and that way it tests the performance of the algorithm. KCML had to deal with noisy Go annotations, which don't provide a perfect ground truth. So more positive genes are required for the classification. And another thing that we think is the downside is the fact that it missed new functions with a new phenotype, because functions are learned according to similar phenotypes, but aren't found by the algorithm. All in all, it was a pretty exhaustive research and with a lot of biological relevance that can be easily reproduced. That's it.
Okay. So first, it was much better than your rehearsal a few days ago. So yeah. Questions? Did you guys understand what was the key point here? I mean, what was special about this? What they did?
It took a few reads of the paper, but eventually we did.
No, I was asking about, no, I know that you understand. I was asking the other students in the course.
Hi. Actually, I think that one limiting factor of this method is the fact that not always knockdown of genes causes phenotypic response.
Yeah, so this is, yeah, sorry, go ahead, Bob and Lena.
I didn't understand the question, I'm sorry.
This algorithm is relies on the fact that it can distinguish between phenotype and find a signature phenotype for a couple of genes that are associated to a certain function, like cell cycle. So if you don't find a certain phenotype, when you knock down a gene, you can't connect. It won't go into the classifier, I think. Or the performance of the classifier won't be sufficient to participate in the whole algorithm.
Okay, so what you are saying is that where there is no difference to the original or the one without the knockdown cell, then it doesn't go into the process at all. Did I understand correctly?
I think it goes, but maybe it considered as noise.
I mean, maybe I mean, I think it's an advanced question. I mean, I think let me. So I'll ask you a question, Lena. How is a gene linked to function usually in cell biology?
A gene transcoded to RNA, which
no, Lena. I was asking Alina.
What were you asking again?
How is, I mean, okay, so maybe I'll take one step back, okay? So what the authors did, they went to an ontology. What is an ontology? Actually, I don't know the formal definition. It's actually very important to something else that I want to tell you today, but it's kind of a convention on names and terms and links that are in a specific domain. Ontologies are very important to allow us, if we think about data integration, building a common language is very important, because I want to go to one data set and then to another data set and combine them together and make I cannot do it if I do not have a common language and a common set of rules and terminology. So ontologies is one of I was involved in building an ontology for cell migration. I think it was, and there was you need to define how was it called? So to define the terms, right? To define terms and then define the links between the terms, and everything needs to be very, very formal. So these ontologies, the gene ontologies, are linking specific genes. So gene A, gene B, gene C, to a specific function. So something in the cell cycle, something in, right? So things like that. So, and this is what I think the nice thing in their paper. I mean, the key idea in the paper, that they are using the ontologies to build classifiers. So they take all the genes that are involved in some function, let's say positively, and all the genes that are negatively involved in some function. Cell growth, right? So big cells or small cells, whatever, or cell defects in cell division, some kind, and then they train classifiers. And then in principle, even if you do not have the ontology, the link in the ontology between the gene and function, you'll hopefully be able to capture it from these classifiers. So I'm asking you, Elena, again, Elena, not Lena, not Nella, Lena, how, I mean, these ontologies, how do they define a link between a gene and a function? Do you know? I mean, it's not a tricky question. I'm asking if you,
I mean, one of the methods, I don't know to answer it professionally, but I think when you find a gene and you don't know its function, you usually use knockdown. But as I understood from Lena's and Bar presentation, the expectation from this model is that it will have a phenotypic result of the knockdown, and it's not necessarily true for many genes. So as I said before, I think it's a limiting factor of this method.
Okay, so this is a limiting factor of everything in phenotypic screening, because if you do not have a phenotype within the cells, you know, the basic assumption is that what you're looking at, what you're looking for, you'll be able to capture that in the appearance of a cell. If this hypothesis, if this assumption is not true, and of course you need to be very careful about it. If it's not true, then on the methodology of, you know, morphology screening is not, phenotypic screening is not, you know, it's not going to work if you don't have this assumption. The assumption is not always true, I agree, but a lot of time it is. And the thing is that the classifier is actually what is nice here, that the classifier learns what are usually there is a correlation. And if a gene knockdown causes multiple effects within the phenotype of a cell, the classifier is actually going to put weights on the features that are actually most important for that are most important to link a group of genes to a group of functions. So if a gene knockdown is going to have like multiple effects, multiple phenotypic defects, it's still going, the classifier is still going to know to put its attention on what is the morphological features that are correlate to the functions they're interested in. Because
I agree. I agree. It's very sophisticated, but if it doesn't have a phenotypic result, you can still check the RNA transcription, and you can check what is the effect on the pathways of the cells. And it's, I mean,
yeah, but Alina, this is not specific to this paper. This is, you're saying, what you're saying is true for everything that relates to cell profiling or cell phenotyping. Basically, you're saying if cell phenotyping might not work. Yeah, sure, but I mean, it's no argument about that. It's not a silver bullet. It's not going to kill everything, right?
I think that the minority of genes will have phenotypic effect, but this is just a guess.
Yeah, but also they are going to have. They are also going to have to be involved in specific functions. They are going to have, you know, very low probability of being involved in a specific cell function. And usually cell function is linked to cell appearance. I mean, in a lot of yeah, I mean, basically, eventually the cells need to, you know, the cell function is something that the cell does, and in order to do it, it needs to organize its internal structure, to, you know, to have some proteins activated and other inhibited, and this changes the morphology. So actually, I would argue that cell function is a functional level readout, and looking at morphology is you know, is a very powerful readout when you want to actually test cell function. You're a bioinformatician, so it's good that we're arguing because, you know, it's exactly, you know.
We're not arguing, I'm just telling
No,
you.
it's good to argue, otherwise it's boring. I'm happy to argue. I don't want everybody to agree with me. It would be boring.
I'm not necessarily agreeing with you, but I'm not arguing.
Okay. Okay, so, okay, I think, okay. Anyway, I think it's a very interesting discussion, and I would like to have it more with bioinformaticians because it's and systems biologists. I mean, actually, it's what should you measure and where is the? I mean, you should measure everything and in every scale because you don't know where you're going to find the phenotype. But yeah. Okay.
Again, it's a very cool methodology. I think it can be. I mean, people can learn from it, but for many genes, I don't know, maybe most of the genes, it not necessarily will show a phenotypic result, and then you won't be able to classify.
Right, but most of them are not involved in this type of functions. But when you look at the function, most of the genes are not going to be involved in a specific function, right? So yeah, I mean, I think that your main thing is that no phenotype, no. So this is true for in general for cell screen, for phenotypic screening. If there is no, if your gene knockdown is not inducing a phenotype compared to control, then phenotypic screening is not going to be able to do the trick. Now there could be two issues here. One is there might not be a function, right? I mean, the gene might not be involved in any process that relates to the cell appearance. And second, we might not use the right readouts. Maybe we are using cell painting with the wrong molecules labeled, or maybe, you know, so.
That's true.
So
about this paper.
One second, Gil, I just want to say one more thing here. So one of the big issues in screening, when you look at industry, when they do this kind of screening for drugs, etc., they put a lot of effort in building the right, and in academia also, putting a lot of effort into building the right system. So what is the right system in screening, in drug screening? A system that will reflect what happens in the physiological setting, right? So you want to take a cell system, a cell line, or a cell from patient that reflects a real cell in the human body, because you want to be as similar as possible. You want to label organelles that are most relevant for the disease, right? That you are in the, so if you know that the disease is causing defects to the mitochondria for an organelle that is called the mitochondria, then you'll want to label mitochondria, right? So you want to build your systems in order to measure the right things that will allow you to not fall into this hole that Alina mentioned. And this is a big deal. This is one of the most difficult things when doing this kind of research and drug screening. Yes, Gil, sorry.
So maybe in order to bypass what Alina just said, maybe they just selected the right genes that they know that going to have a phenotype from changing them, from knocking out them. Maybe they just selected the right genes to check. I mean, it's not some arbitrary selection of genes.
Yes, they did it in the. So this is what they got from the ontology. The ontology told them that genes that are involved in a specific function. This gene is involved in this specific function. So
this is for the functionality. No, I'm speaking about the knocking down and see the phenotype. Maybe what Alina said.
No, they didn't. No. This would be cheating.
That's right.
If they want to say a method works, then they need to, select. You cannot exclude based on criteria that there is no phenotype.
You can, but it's not good. But
yeah, it's not going to be a discovery machine anymore. It's not going to allow you to discover anything, so it's not very helpful. Okay. What else? I had something else that I wanted to say. Can you go back a slide or two? No, another one. One more slide back. Yeah, this. So what did they do here exactly? I mean, the KCML I understand, but what did they do? What is K-means? K-means is clustering. How is it related to?
They tried to classify here. They tried to classify the phenotypes, the different types of phenotypes, and see how the signature classification works. So if they classified, if we go back a little bit here, we can see that they classified to 2 different types of signatures. And then they say that the other types of clustering methods that they saw, they don't really cluster that well the phenotypes, while their algorithm does a much better performance of clustering.
So for example, when they do, it's not clustering because they do classification, but let me ask you if this is what they did, for example, for, let's say, K-means, okay? So they had positive genes and negative genes for a specific function. They took all the data, all the phenotypic data. They did the K-means with K equal 2, so they did K-means for 2 classes. And then when they have a new observation, they ask for which class is it. Is this what they did? So kind of they cheated because they did here things that are not very, for K-means, for example, it would be unsupervised clustering and not supervised.
Yeah, so they didn't really go into depth about the whole process, which is one of the things that we said that was like not really good about this paper because it was more biological than computational. So a lot of the metrics and of the different research processes that they did were not in the paper itself. But from what we understood, yes, they took they clustered the different signatures with the positive genes and the negative genes, like the random genes, and then they tried to classify the new gene to where it goes. And with the K-means and the self-organized map and the Pearson correlation, they saw that it just randomly classifies it, because when you get an AUC of 0.5, then it's basically random classification.
Okay. Okay, sababa. Any other questions, thoughts? Okay, so thank you. I'll take over now. Come on, what's going on? Okay. Okay, so I want to quickly summarize, and then I want to talk about another last topic and do another last topic that I think is relevant for where I'm going in the summary. So actually, what I did here most in this short, relatively presentation is still slides from the reshuffling some slides from the first lecture. But now you understand. Now maybe I hope that you understand and you have more context. So the course theme: review state-of-the-art in visualization, processing, integrating, and mining cell image datasets, deciphering complex patterns and turning them into new biological knowledge. It will include a variety of computational approaches applied to diverse bioimaging data. Okay. Quick Q&A. No need of background in biology. We communicate in English. I think it wasn't as bad, right? As you would. I mean, what do you think about these two things, about prior knowledge of biology and about the English thing?
If I can say, it's kind of hard not to have a biological background in this course, because it's very interesting, and once you go into everything, then it's really interesting, but it's kind of hard to follow during the class if you don't have the biological background.
Can you be specific about one example?
One doesn't really come up to mind right now. I can send you later if you want.
Okay.
An email, but it was kind of hard, like the whole processes of the cells and everything, and I'll send you later on. But
okay.
I think that for the classes, biology, I don't have any biological background, and I think that for the classes it was okay. Maybe from time to time, maybe a minor thing, but it was overcome later. Reading papers is a different issue. When I try to go to a paper that we speak about, it's Chinese, it's not English. But okay, we are in class, and we study it in a different level than the actual paper. And my feeling is that in class it's okay. It's for at least I managed to understood. About the English, I'm not sure that I would insist on it, but it's your choice, it's your call, the English.
Okay. Other thoughts? I mean, I was trying to make accessible English, right? To make accessible the biology. So I tried to ignore genes names and stuff like that. And in principle, whoever was in class, I was encouraging asking questions. Reading paper, I agree. Reading papers is a different story because no one is mangishim for you when they write their papers. You're not the obvious audience, right, for this type of papers. So, but in class, I don't know. Maybe I should emphasize more that people should ask questions if something is not clear, because this is something that I'm trying really hard to make accessible. And if I'm not doing a good enough job, then I need to know. This is why I was asking for specific examples now.
I think that if it's the connection between the biology, the no knowledge in biology, and the fact that the course is in English, because like there are some words that you need to go and search and figure out, and then it kind of...
Yeah, but then I was trying to I mean, probably I didn't do a good enough job in that, but I was trying to promote you guys to participate, and then part of it is asking questions, is something. But now thinking back, there was not a lot of questions about terms that were not obvious, so maybe I didn't make it clear enough that I'm happy to stop the lesson and explain, and if something is not clear. Anyway, I'll take that, I'm writing it down, and I'll make it into consideration for next year. Okay, let's see. This is not a deep learning course. I think I made it clear this year. Last year, there were a couple of students who thought they're going to learn hands-on deep learning. This is not the idea.
Although there were nice deep learning tricks in here. For example, in your work for choosing the right bit, the right argument for checking its influence on the target. I think it's in your own paper, right?
Yeah, but
it's a nice trick.
It's not a deep learning course. I'm not going.
No, it's correct.
Why one architecture is better than the other, and how to do exactly. I'm not. It's not.
That's okay, but there was a trick. We learned the trick.
Okay. This is a tweet that I did like 2 years ago, which I think I have an answer now. I mean, since so I was curious about, because it's the first course of its kind. I mean, there is no other course like it. I'm not aware of any course similar to this. So I was interested to. I mean, was curious to see if computational students would be interested in this type of, very esoteric for you guys, for applied computer science department. And yeah, so this was my personal motivation. So I started the course with this movie and a very simple microscopy from the 1950s showing, you remember this movie? Or do you want to see it again? No? I'll show you again then, and I need to share I like to watch it every time. So let me see, share sound. movie from the 50s where bacteria, neutrophil, a white blood cell is chasing a bacteria, and these are red blood cells. And you can see, I mean, even in such a simple, with such a simple microscopy and such a simple setting, you can see very complex cell functions that are taking place. And understanding how these functions, and you can see, I mean, the neutrophil needs to process information from the environment, organize its internal organization in order to chase the bacteria. And even a bacteria, a simple creature like bacteria, can identify, can sense that a neutrophil is chasing it, and decide to, to move into different directions and organize its internal structure and do everything. So such a complex information processing and decision making and sensing is something that we are trying to understand in cell biology. The only way to see it is microscopy. I mean, there is no other, to see dynamic events that are happening and understanding how they happen, there is no other way to capture these processes. And they are super, super complex. A lot of things are happening here. And in order to understand how these functions are happening, and we need advanced computational tools. So seizing, believing, hundreds of years, we're looking under the microscope like here, and coming to learning about the world and coming to conclusions. And in the last like 20, 30 years, it's not enough anymore. We need also to put numbers in order to show that. So previously it was here, look, here is a nice picture of, and based off this nice picture, I would say that big cells are, I don't know, more something than small cells, right? Now we need to put numbers in order to prove that it's systematic and real. And to do that, there are a lot of bioimage analysis tools that are out there, proprietary or open source, a lot of open source going on out there that are giving us the tools to actually do this type of things. When we know what we want to measure in advance, which is usually we want to measure something about cell trajectory, about the cell size, about some organelles within the cell. We have these tools which allow us to actually measure something specific that we want to measure, and then we can put these numbers. The problem is that a lot of the cell biology is too complex. Our eyes are not good enough are not trained and cannot, because the processes are so complex that we cannot see them with our eyes and say, I want to measure this and that, and please build me a tool to just measure it, what I see with my eyes. And here it becomes exciting. So I'm not interested about if a biologist comes to me and says, I see this phenotype and I want to measure it. I'm not interested. I want to measure things that are not seen by eye, that we cannot assess by eye, and we need the numbers in order to understand them. So the idea is to flip the order and do analysis of the images, get numbers, and from the numbers come with a biological hypothesis. So come from the data exploration, come with an hypothesis and ask the specific question. And in order to do it properly, we need to have domain knowledge. What is domain knowledge? We need to understand the domain, the biology, not all the biology, but the questions that we are interested in answering. And we need to know what to look for. We need to know how to look for it, what experiments should be done in order to look for what to understand, where to look for it in the images, and how to extract this information computationally, quantitatively. So we need to be, in order to contribute to the field, we need to have domain knowledge, both in the cell biology, at least specific to the questions that we're interested in, and in the computational tools that we're able to use in order to decipher, to solve these questions. So if you think what computer vision and data science have brought to cell biology and microscopy, first is automation. Even until today, there are poor graduate students in biology departments that are clicking on single cells and events, and it's still, I mean, it's fine. We still need to do it from time to time, but in principle, if we automate processes, if we have a lot of information, and automation will allow us to do this in a way that sometimes you just cannot do it. You have hundreds of thousands of events, millions of events, and you have the mental health of the students that cannot spend all their masters or PhD in click or postdoc in clicking events. So So this is very important. Completeness. What is completeness? Completeness is looking at the whole distribution and all the variability within the data. So previously there were, oh, this is a cool cell. Let's quantify this cool cell and that cool cell. But then we introduce biases. We're not really understanding the whole, everything that happens within a population of cells, but we are focusing on very specific cells that capture our eyes and that we select them based on some biases that are not necessarily relevant. So not necessarily reflect the full complexity of the data. So completeness allows us to quantify all the data. And then it also allows us to focus on interesting subpopulations. So for example, if you think about cancers, there is a very small subset of cells that can cause the real damage eventually and go and metastasize and do the real damage. Then we need, we need to be able to not to focus on a specific subset of cell based on our bias decision, but be able to look at the whole distribution, and then maybe we'll find something in a rare subgroup of cells. And the most exciting is finding invisible patterns. So being able to find something in the data that the human eye cannot capture. So automation and completeness in principle, if you had infinity cheap manpower, you would be labor, right? You would be able to do it. And finding the invisible patterns, this is the data exploration. This is the real data science that is in the field, trying to really understand what are the patterns that are involved. So this is what I wrote in my lab website, but it's also relevant for the course. The motivation is usually fundamental question in cell biology. Now there are also a few more applicative, but in principle it's basic science. And what we produce or what we're interested in is one, biological insight. And if we develop tools, and we develop tools that were not developed before, so tools that measure something new. I'm not interested in doing a better segmentation algorithms than other segmentation algorithms because it's not going to allow me to answer a new question. So it's just going to show off that I have a better algorithm. I want to be able to answer a question. So if there is no tools to measure something in order to answer a question, I'll build it. I'm not doing anything. My students will build it. I'm just writing all the day and speaking. But the students in the lab will do it, of course. And if there is an existing tool that will solve it good enough for my purposes, so it's a very engineering-like. So it's a combination of engineering and data science, right? Using whatever tools you have and mining the data to find these hidden patterns. So the course objective was to bridge this gap between cell biology and computer science or data science. The nails are the important and exciting problems in the biology. I mean, all the world now appreciates that biology is like the coolest place to be in terms of science. You're seeing, even in the university, you're seeing mechanical engineering department and industrial engineering and data science departments and almost every department is recruiting someone who will do biology because it's very important and very exciting understanding life and solving these basic questions. And we have all these amazing tools that have not done the aliyah to cell biology, right? Are not used or not utilized yet in cell biology, in microscopy data specifically. So, okay, so my personal opinion, so some computer scientists are doing the whole way to biology, and some of them are doing amazing jobs. They are really amazing. I would never be able to do something like that. The limitation there is that a lot of time even if you're fantastic, you're not going to get you cannot do everything. One person cannot be great at everything, right? At building a very powerful experimental system and doing top-notch computer science. It's just really hard. And the second thing is that once people decide to build a wet lab, a lab that does heavy experiments, it means that they are invested in the experimental system. So they build experimental system. It takes years to build a powerful experimental system, and then the focus is there. And the advantage of being a data scientist without this is having flexibility. So we can work one day on one problem and another day on a completely different problem. We collaborate with a lot of biologists that have these fantastic systems that they built by themselves, and this is something that I think is very exciting. It's not a traditional way of doing things, so it's still, you know, it's not very common, but I think it's worth trying to do it also to do it this way. Here is a quote from a very prominent review about bioimage analysis from 5 years ago. And you see here that the focus is replacing human labor as much as possible with computer calculation so that biologists can focus fully on formulating high-level hypotheses and designing increasingly sophisticated experiments. And this I didn't like, because why? Because I want to think as, I mean, data scientists think a lot like, they do data exploration, can think a lot like biologists. We can ask the question based on the data. And this question cannot be, I mean, I want to think of myself as trying to be a biologist, so trying to ask the biological questions. And it's not necessarily my collaborator who is going to ask the biological question. It could be the data scientist that is asking them. So what is the problem? I think that this resembles a problem. It's more than just words, a paragraph in a paper, because I think that most of the computer scientists who do the step toward biology are building half a bridge. So they want to work with biological data, and they work with biological data, but what they do, they build tools for obvious tasks that does not require a really deep understanding of the domain or of the biological question. So usually most of the people who do this step, what they do, they are tool builders. They are working at improving segmentation algorithms, improving tracking algorithms, coming with new algorithms for noise reduction, and I think that it's great, and this community is fantastic. They're doing amazing things, really. I mean, and the tools are very important, and I would even say that they are more important than a specific biological discovery. Being able to develop tools that a lot of people will use is more important than anything else. For me, it's just, I think it's a little bit, it's less exciting than discovering something new. And I tried to, with Megan, friend Megan, we wrote together a perspective where we tried to characterize that, and we defined a low level or signal-driven tasks and high-level or biology-driven tasks. And in the low level, you can see registration, tracking, segmentation, template matching, deconvolution, all kind of basic or not basic image processing tasks that can be done with different level of complexity, and require some domain knowledge, for example, in the optics, in microscopy, but are not necessarily require you to dive deep into the biological question. And the green parts here are more into what tools you'll need to use in order to actually answer biological questions. These are like what we call high level, and this is more about the data mining and data exploration. And I think that as a community, I mean, we need to bridge this gap, and the way to do it is to think, I mean, at least the way that I think that doing that is doing this interplay between developing computational tools and then thinking of what problems they can solve. So building hammers and then looking for nails. For example, the interpretable machine learning gives that you mentioned, we did it for one application, and now we're looking for other applications. And we're building tools to measure how information propagates in multicellular systems. I showed you that. And now we are looking for more and more applications of multicellular systems that communicate with where cells communicate with one another. So we can use these tools in order to answer new questions. So yeah, so the idea is to go beyond bioimage informatics, beyond the tool building, and try to drive computational folks to be actually involved in asking the biological questions. And this is a little brainwashing maybe, but this is how, I mean, this is the reason why I came to a computational department after my postdoc, because what I wanted to do is open this world to computational people that will do the extra step beyond tool building. And then it will be a better world where biologists will have new tools and we'll have new problems and everybody will be happy and blah, blah, blah. Okay. Let me finish this presentation. You know what? Let's do this if you agree. So we're going to finish today at 4, not at 5. I forgot to tell you because I have a seminar that I want to attend, and I thought, I mean, we have this August meeting anyway, and we have so I think it's fine if we lose an hour. I mean, it's I mean, I can talk for another hour, but I want to hear this seminar. So you get שעה חופשית. So תיקחו כדור ותלכו לשחק או משהו. So okay, so let's do בלי הפסקה until 4 and then or 5 דקות לפני 4 and then we stop. בסדר? תחזיקו מעמד. Because I do want to get somewhere here. I want to brainwash you completely, not just partially. Okay, so the course description. I wanted to show you. I wanted it to be a very broad course. So I wanted to show you a lot of biological applications and a lot of computational tools, and not to focus on one specific domain. And not going deep into nothing, no biology, and no, we didn't go deep into any specific topic, computational topic. It was all very on the redud, right? Because I wanted to open you to this amazing world, and also I wanted to allow this course to be suitable for a wide audience. So I was so people who are not completely computational or not completely biological could take this course. Computer techniques that we've shown, some computer vision, machine learning, time series analysis, networks, etc. And of course, I was biased toward the things that I'm specifically interested in. Also, the course is biased. I mean, I showed you stuff that I did or in my lab, which is, you know, it's probably not the best example of probably I could have worked harder in finding the best examples for data science in cell imaging, but I'm biased and I'm most excited about what I do. So sorry. Grades. Paper presentation is 20%. Final research project is 80%. I have, you know, we're right, and we did already the paper presentation. I have my notes on each and every one of you. Final project groups. I met already with most of the groups. I was very impressed by many of the projects. Very creative, and I'm very מאוד מבסוט by the ideas and by the way of thinking. Now we need to see what comes out of that. But so far I'm happy, and in the process of iterating, getting approval before going on is good. I think I'll keep it because last year it was not as good. Yeah, I wrote here some. So the project types are, in which most of you took, is data mining, and not necessarily in public repositories. And in public repositories, mostly in datasets explored in the lab. Collaborative bioimage analysis project, we didn't have this year. I had one request for that, but I didn't deal with it so it was my fault that I didn't deal with it in time, and it's not relevant anymore, I think. And then tool building, there is 1 or 2 projects that are involved in trying to develop new tools, or maybe a little more. So we are going to have a project presentation day in August. You need to submit your project to me a little bit before that. The written report is very important for you and for the grade. I need to understand what is the problem, some background, very brief background, right? What is the context in the course, background about the problem and the data, what you did, what were the results, and what was the interpretation. I'm going to limit the length of the report. I'm not sure how long, maybe 7 pages or 5 pages or something that is not, I don't want you to write a lot. It's also, I think it's a better exercise in writing, because now I'm reading, I have for tomorrow morning to read 3 reports. Actually, I read one of final projects in the department. Each one of them is 30 pages. And it's really, students just dump a lot of text out there to fill in space. It's not really, you don't learn from that how to really write. Writing is you need to, I mean, it's also, I think it's good exercise. Beyond that, it's better for me to read 5 pages than 30 pages, right? But I think it's really better, you know, it's not, you're not going to do a huge project, and there is no reason why writing it in 30 pages. You can focus it to 5 pages, and it would be a very good exercise. The average grade is going to be 90 with some distribution in the course. So this is, I mean, this is how I'm going to do that. So I'm going to define a מפתח ניקוד. So eventually the ממוצע is around 90. Yeah, so what did we learn and what didn't we learn in the course? So the red is what we didn't get to learn. Natalie came and gave us a fantastic talk about cell biology and microscopy. Genevieve about bioimage informatics. Then we had a long, like maybe 3 sessions about deep learning in microscopy, and also a lot more than 3 sessions, but there were a lot of student lectures. I would say 3 sessions about high content single cell phenotyping. I think these were the two topics that we got to see the most of in this course. Then we skipped these topics. Advanced representation of cell shape, intracellular organization, trajectory data. Atlases and public data repositories, I'm actually hoping, so I have my other presentation. I want to show you a little bit about it because I think it is important. Because when we talk about data science, and if you think about, for example, bioinformatics, the big revolution came with the data. When the data came, when the huge data sets came out and atlases came out and made the data publicly available, then suddenly the computer scientists came in and started contributing, and now it's a whole field, right? I mean, now it's a huge field of bioinformatics. So I think that making data publicly available and making these big, massive, and comprehensive data sets is super important for the field to evolve and move forward. I showed you some work from my research on information processing in multicellular systems, computer vision and cell imaging. We had the talk from Tami from BGU from electrical engineering, and then we had Tal Shai. I need to say Tal Shai, not Shai Tal, because Shai Tal is a different person. About importing ideas from systems biology. Let me change it. I think it's, so I don't, and I think I'll make it, I'll be optimistic and I'll think that we'll get it today. And we had Paul talking about a very exciting venue of integrating visual data and omics data. And then we didn't have time to speak about some other topics, which are specific examples of how we can learn new biology from old data, automating microscopes. So now today you can hook into a microscope and build in machine learning models, for example, that will learn how to make decisions, how to allow the microscope to make decisions in real time. So, for example, an application for that is imaging. If you want to imaging a developmental process that takes a lot of time, the microscopes, you need to autofocus. You need to focus the microscope during the day. You need to optimize different things, and you can build automated tools to make this decision on the fly. Simulations, I didn't have time to talk. Medical imaging, I actually didn't want to talk. 3D image analysis and visualization. Okay, I think an important point here is that the field is lagging. So it's an opportunity for computational people. It's really an opportunity because we can do a huge impact. I mean, if you are going into, when you select a research problem or a research field, whoever in the class is thinking about, for example, doing a PhD, you don't want to go to a field that is already super mature, and you can, all your contribution could be very, you can contribute only something small. You want to go to a field that is emerging, that there is a lot to do there and a lot to discover and a lot. So think of, I mean, it's not. This is one of these fields, and if you are considering to go to a higher degree, then please take that into consideration. There are other fields, of course. I'm not. I just want. I think it's important to think about it. One thing that is cool about the field that is very different from computer vision, because in principle, tool-wise, computer vision is using more fancy tools that I'm using, right? The state-of-the-art in computer. I'm not. I was in a computer vision lab in my PhD. I'm not anymore. I'm not מעודכן. I'm not fully מעודכן in the literature, for example. Updated in the most recent literature because I cannot, because there are too many things that are going on and too many fields that I'm trying to keep track of. So the fancy tools are there in computer vision, but one of the most cool things in this field and that is better in this sense than computer vision. In computer vision, the human vision is the ground, is the holy grail. Computer vision systems want to get the ability of a human person. And you can see for the application what they do and how they try and getting better and better, and it's an amazing field. But in data science, in cell imaging, we're actually doing more than the human eye. We're capturing patterns that the human eye cannot capture, which for me, it's very exciting. Yeah, so it's an opportunity. I have twice the same slides. Okay. Yeah, one of the difficulties in using cell imaging data is the variability. Each lab is doing different experiments, and it's a very messy data. It's not like when you do computer vision, you can go to, when you develop an algorithm, you go to a data set, and all the data sets, hundreds and thousands and tens of thousands of people already played with this data. The data annotation is fantastic. It's really easy. And the data is organized, and if there were outliers, they were excluded. Everything is clean and ready for you to come and try your algorithm. In cell biology, it's not the case. Even, usually when someone creates a huge data set, they do some analysis on it, but they are the only people who look at this data. And it means that when one of us is trying to play with public data, or data through collaboration, it's going to be messy. I can tell you of many times that I went to my experimental collaborators and told them, hey, what went wrong in this and that experiment in a specific day? And because the data told me, right? Because I know how the data should look based on other experiments, and suddenly I see something that is completely wrong. And then they look at their notebook, they are very organized in doing the experiment. They, oh yeah, we did something different here, or so yeah, this is another challenge. And now I want to really quickly to show you some of the highlights of the course, and of course with nice images and movies, and then move on to the last topic of the course. So content-aware image restoration. Here you see the raw data, and here you see how the data is improved with the care analysis. And basically, the idea here is that you want eventually to track these cells, these nuclei of cells. And with the original image, you can just not do it. And now the nucleus are crisp and clear, and your analysis could actually process this data and do post-processing within it. I think one of the coolest ideas that I was really surprised when it came out and that this idea worked is in silico labeling. It's amazing that you can create from these images, you can create these images right below. It's unbelievable that this information is hidden and that the computer can learn to map these bright field images into the organelles. I mean, I'm still, it's fascinating, I think, and it's the potential. There is a lot to do here. I mean, there is a lot of potential, and there are a lot of caveats. It's not pretty as it looks, of course. It's if you really want to use it to learn something, you need to be very careful, and we're trying to do some steps in this direction. Of course, one of the highlights of the course is my paper about the melanoma, the morphing of the melanoma cells, where we found directions in the latent space based on classification that are pushing cells from one functional state to another functional state, and then we pushed them. Now we're trying to use similar methods for other applications, such as in vitro fertilization. So trying to do similar things for embryos in a plate, and trying to see if we can predict based on a subtle phenotype that cannot be seen by eye, whether an embryo is going to be a good or a bad embryo in the future. Discovering drugs in a high throughput, we had a lot of discussions about that. I showed you also work from the lab about the information processing in multicellular systems. I think this is for me, this is my signature. I mean, this is the thing that these are the projects that I'm most excited about. Trying to understand how information propagates in multicellular system. I'm hoping that in 10 years from now, it will be attributed to me, okay? So the computational tools to measure that. So I'm most excited about the potential of asking these questions. And the idea is basically to build a network of how information propagates. And in order to do it, we are looking at time series of dynamics events that could be a lot of things, and we are trying to build networks that characterize how information propagates within this system from one cell to another. So a few examples of projects that we have started in this direction. Here you see cells, fibroblast cells in a fibrous, like a jungle. The red is a jungle and the green is our cells. And you can see that the cells are communicating. How do you see that they are communicating? They are pulling on these ropes and they are creating these denser fibers that they communicate. And Assaf from the lab create tools to actually measure this process and show that cells that they communicate, they have a very unique signature. Propagative cell death, it's also a communication process because a cell is dying based on a decision of a cell that died before it. So it's a community effect. I mean, it's a peer pressure. I die and then they say, Achi, kidai gam lecha lamut, and then he dies also. Although he was very tough before that. But when I told him you should also die, he was very convinced. So anyway, we are building tools. Shaya is now building tools, computational tools, and also using simulations, agent-based simulations to actually measure these processes. And
by the way, all those questions about communications are not yet known to biologists. I mean, it's not something that we have biologist answers. I don't know, by checking the proteins or the hormones or whatever moving between them. They don't yet have the answers.
So people are talking about communication, and the communication also, it's confusing because you can have it in multiple scales. For example, if you look in a tissue and you're looking at two cells and you are so for example, even a field of spatial proteomics, right? That is completely static. You're looking at the proteomic states of single cells within a tissue, within the context of a tissue. And even then, people are talking about communication when they see enrichment in multiple cell types that are together. So people are talking about communication, but it's in different contexts. In live imaging, people are talking about synchronization or coordination, or which is basically also communication. But usually the ways that people study these systems are they say, okay, we're defining, for example, we're defining a measure for communication or synchronization. For example, a correlation between something within the cells, right? Correlation between a molecule within the cells or between their migration or whatever. And now we perturb the system. So we take out a protein, we knock down a protein, and now we measure the same thing and we see how we change. And if we see that, for example, the collective migration, like I showed you, is moving slower, or the cells are moving in less coordination, then we say that this molecule is involved in cell-to-cell communication. My idea or my philosophy is that this is a top-down view, and we need to learn it bottom-up. We need to go from the single cell and understand how information propagates within the system. And in order to do that, we need tools. Because a lot of things, because in order to understand the system, we need to understand how its components are building together modular structures that eventually will build a collective. And because of the heterogeneity, there is a lot of variability between the cells. And it's not enough just to look at the population level. We need to understand what happens at the single cell level, and from that being the bottom-up model. So people are talking about communication, but they're looking at it in different directions. And what I'm going to, for example, I'll give you specific examples, okay? In this system, okay? People talked about mechanical links between the cells. How did they measure it? They measured it by saying, okay, there is a band. You can see here, oops, no. You can see here a band of lighter red, right? So this is, and they said, okay, this means that the cells are communicating, are mechanically linked. And if they want to measure it, they measure how bright the pixels are here in this band, and they say, okay, this is how they're connected. But when Assaf developed these tools, Assaf Nahum, my student who graduated last year, he said he actually built tools, and he found that it's not correlative. It could be that the cells are better communicating even if they don't have a band. So it could be that these two cells are communicating even if they do not have a band. It could be that, so he actually was able to put numbers on the communication that were not, and he found that cells that do not have bands are communicating. He found that even if you reduce most of the contractility of the Yeholet Kivutz and fourth generation of the time, even then the cells are communicating. You need a little bit contractility in the cell, but even if you have very little contractility, you're not going to get any bands like this, like you see here, because the cells are not generating enough force to actually deform the space between them, the fibers between them, but they are communicating. If they are sensing each other, I mean, so you need these tools in order to start asking questions, starting to really understand that. For example, collective cell death. You'd be surprised on what tools there are there to measure these processes. The tools are completely, they are not considering at all the spatial location. So the tools that are now to measure cell death look at the percentage of cells or number of cells that die as function of time, and then they characterize the graph. So they ignore completely all spatial components. So even a simple tool that students from the lab, it was Chen and Liran and undergraduates actually, and Tom, that builds very simple tools to just show that to measure these propagations that you see here on the left, to show that it's not random, that it's propagation, and put numbers on it, which is very simple. There is nothing complicated about it. It went to a journal that was accepted to like top-notch journal. It did a lot of it was made Ynet, etc., which is probably more exciting, right? No, but it's really, I mean, it made a lot of noise in the, and it's just a field that is so behind that there is so many, and the field of cell death is specifically very behind, but I'm not going to tell you why now because I want to go to other things. So did I answer your question, Gil? Short question, long answer, sorry. This is me. This is pretty cool. I just want to show because it's a cool video. So what you see here, two populations of cells. You see the two populations. You see that these are dying. Oh, you can see this means that they're dying. And only after all of these died, it starts to propagate to the other cells that are dying. Look here. So it means that these cells at the bottom right side are much more susceptible to this. They are much more and they don't need a lot of convincement to die, but only after all of them die, there is enough convincement to the other side, enough pressure to the other cells that tells them now also to die, which is a pretty amazing experiment. And we have no idea why it works this way. We have no idea. Or by using these tools in Amos's project, we can actually learn that the cells build a communication network. They have different roles in the network. They have memory. They begin by learning their local environment. Through time, they start learning about the global architecture of the network. It's pretty amazing to coordinate their behavior. It's pretty amazing if you think about it. And we must develop these tools in order to be able to describe these processes and then understand what molecules are involved in these processes.
נראה שהווידאו נתקע.
כן, נראה שנפל לאינטרנט.
נראה
כן.
שנפלים.
הפסקת חשמל או אינטרנט או משהו, כן. למישהו יש את הקונטקט שלו, ליצור את הקשר?
נראה לי שיודע שהתקשורת שלו נפלה.
כן.
אגב, אם מישהו רוצה להציג את השיר שלו, ואז הוא יפרסם את זה, זה יהיה נחמד. You're on mute.
אסף, אתה מיוט.
אמרתי שלא, I don't know how long I spoke until I realized that I'm speaking to myself. מתלהב לעצמי. Anyway, I assume it's somewhere here. So, anyway, יאללה, מספיק, הלכה לי ההתלהבות מהפסקת חשמל הזאת, אז let's go on then. This is what I told you, event-driven microscopy. What you see in the...
אתה לא משתף את המסך עכשיו, אסף.
Oh, no. עכשיו זה בסדר?
עכשיו טוב.
אז את זה ראיתם, נכון? You saw this amazing cell in 3D, right?
זה עצר בדיוק בהתחלה של זה.
ממש התלהבתי פה, ועכשיו הלכה לי ההתלהבות, אז נראה לי אני אעבור הלאה. The systems in cell biology are becoming more and more complex from different aspects. This is the summary, and one of it is 3D, and we need to build tools and to ask these questions in 3D. This is one example from done by Megan, my colleague, immune cells in 3D. In what you see here is built is the microenvironment, the CVIMs that I showed you earlier, like the red thing that was in the images, just in higher resolution, so you can actually see it. Anyway, this is one thing that I didn't have time to. Another thing is automated microscopy. So what you see here on the left is a developmental process of, I'm not sure even what, but probably a fruit fly, probably a Drosophila or something. And this development process, look at them. I mean, the organism is changing so many things in 3D, it's like crazy. And in order to image that, for you want to see the whole process, you need to change dynamically things within the microscope, change parameters of the microscope on the fly. And this is a nice paper that I think started these ideas of automating microscopes to actually make decisions on the fly. This is something that I didn't have a chance to talk about. And that's it. I have another slide deck that I wanted to show you, which I will go very briefly because I want to hear your feedback on the course. So this is about reusing cell image data. And talking about, I mean, because I brainwashed you last, with the last presentation about data science in cell imaging, blah, 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 why it's cool, why it is important, blah, blah, blah. But in order to actually develop it as an independent field, we need data. We need access to data. I mean, you see that in every field that opens the data and makes a public big repositories of data available, computer scientists are coming. We're like, זבובים שבאים לזה, כן? We are driven by data, so And yeah, and the idea is that we need data, they'll get methods, everybody will be happy. So how to do it? So this is like a microscope is data generation, this is sharing, and this is recycling, reusing, right? So in order to do it, anyway, I don't have time to speak about it. I have a perspective that I wrote on sharing and reusing cell image data. I want to talk about now the public repositories because I think now is a critical stage for this. So the time is now. Why now? Because now all these huge data sets and repositories are coming out. And I don't think that it's by accident or by flukes, right? I think it's the field is ready. The field understands that this is where they should go, and they are producing those huge data sets, and there are still not enough people who can come and make sense out of this data set. So one data set that they are already described in this course is the JUMP Cell Painting Consortium from Broad Institute and a lot of drug companies that they come together to really build like huge data sets for screening, for phenotypic screening. And actually I saw just maybe this week, I don't remember exactly what day, but there came up, the first data set came out from this consortium. It's a new resource where they have matched chemical and genetic perturbation. So they do genetic perturbation where they know what is the, they do chemical perturbation where they know what is the genetic function. And this can be used and so now you have ground truth and you can actually use it to validate different tools. So I think this is very cool.
You can make research of the data scientist propagation in new fields with the new data.
I'm very specific into cell biology, but you can do it.
When I finish my PhD.
I can do it in cell biology, but it's too early now. So you're going to see a flat line and maybe now a small peak, but you know, maybe we can do it in 10 years from now on cell biology. We can do it on tool building in cell biology, but I'm less interested in that. I told you that I had slides last week about, and these were the slides, not all of them, but partially. So I talked to you about the image data resource. So I found them eventually. They were in some recycling something. I showed you that what they did it was very simple, but it was, I think it was a very nice demonstration of what can be done, where the field should be heading into data integration. So what you see are color-coded, these are molecules that are related to some function, and the color code is different studies, so different data sets. And they integrate the different data sets together to get a more complete picture of the network, the protein network that is involved in cell elongation, right? In a specific cell function, the elongation of the cell. So this is a very simple example of how you could integrate data from different resources to find something, to learn something new, to build more knowledge. And IDR now have tons of data sets, very rich data sets. We need people who will think what to do with them. I mean, I have no idea right now also because I didn't dig into all the data sets there, but I'm sure that there could be cool things that could be done by thinking on how asking question on one data set and then validating it in another data set and then coming up with a new question, but you don't have the molecule. So what? We can jump to a third data set and ask it there. I think it's a new way of doing science that could be promoted. The Human Protein Atlas is another fantastic resource where they basically, they used antibodies and they stained every time a different organelle with some landmark organelle and then a different organelle each time. One cool thing that they did, I told you about, is citizen science. So they took just people from the street and made a nice computer game and got, I don't know how many, but years of human labor time for people who play the game. And actually what they did is annotate images for them, which is, I thought it was very cool. I heard a lecture of this Emma Lundberg, and it was pretty, they did it very, very cleverly, right? So it's very hard to get so many people involved in in doing citizen science. So they say that people are happy. It's a cool game, but people are also happy because they know that they are contributing to society. So they are motivated. So they are motivated by contributing to society, but they need to have fun playing the game because otherwise they'll stop, right? There was a protein localization competition, which is, again, this is something that computer scientists and data scientists really like, because then they compete on which one is better. And this also, so what they did, you have landmarks, which is the nucleus, microtubule, and ER. And then you have a mystery, the protein of interest. And then you want to, the output is annotation of where this protein is located. So you have labels of this protein, you can see that it's located to cell junctions, and you can see that it's a little bit that it's also in the plasma membrane. So the classification task, you get a protein of interest and the landmarks, and you need to give annotations of where it is localized within the cell. And you can use this type of readouts to make new discoveries. So this is actually a very recent paper that came out just this year, just a few months ago. And it's not just using the public data from the human protein. They generated the special data for this project, but now it is available in the Human Protein Atlas. You can see here that's what they did. They did imaging proteomics, which is what they do usually. They also did single cell RNA sequencing, and then they tried to link them together and to learn something new about to build a comprehensive map of I'm missing here my text. Anyway, of cell division and looking at the heterogeneity, etc., etc. Another resource, and now we're going into philanthropical resources, which I think philanthropy is doing here a better job than national funding agencies. I don't know if it's better, but in this field at least, yeah, I would say even better. So you have the Allen Institute of Cell Science, Paul Allen, one of the founders of Microsoft, funds it, and the motivation is building a model of the cell organization, and they have a lot of resources that are available out there. Cells that you can take to actually do experiments in your lab with their cells that they engineered, tools for analysis and visualization, open data, and yeah. And I wanted to go over this paper, which I think is cool, but I'm going to not to do it because it's too complex. And now I'm just going to say that they build an integrated model of where proteins are localized within cells, given a special, given their morphology. So they did kind of morphing. You can take a cell, you can morph its shape, and they would know to morph the organelles within the cell to how the cell would look if the morphology would have changed. And now they wanted to see the variability of different organelles within, based on this limitation based on this knowledge of a cell has a specific geometry, because a lot of the cell organization is driven by, is defined by is at least correlated with its shape, right? So eventually they've built models or average models of cells that you can look at how the organization of different organelles within cells in different shapes. And they also had a measure on how well you can predict the localization of an organelle based on the shape, when you know the shape of the cell. So there are specific organelles that you know to predict very well where they're going to be located, where you know how the cell is, how the cells look like. And some of them you know less well. And for example, one cool application of this measure, I think cool because we did it, Katya did it from the lab, is actually correlate they call it location stereotypy. So if you know the geometry of the cell, do you know where the organelle is localized? And on the y-axis here, oh no, I have the same label here, which is of course wrong. On the y-axis, you have the ability to predict from in silico labeling the organelle. What does it mean? So high values mean that we can, high values here means that we can using bright field images, we can predict where the organelle is located. So you can see here the upper parts here, which means that when you know where an organelle is located based on the geometry of the cell, the architecture of the cell, so based on global properties of the cell, you'll be able also to predict it. So it gives a lot of information to the network, basically, because the network sees the whole cell, and it gives it a lot of help in understanding where the organelle is going to be located. But what I think is even cooler is when you see at the low values here. So there is very low location stereotype, right? There is very low, so it means that the cell shape is not giving us any information about where the organelle is going to be localized. And then you can see here a lot of variability. And this means that some organelles, the network learned where to find them based on local texture properties. You can actually see locally within the bright field images, you can see this organelle. The network sees this organelle, and it's not using help from the global information about the cell structure because it's not related to this property. So here's an example already of trying to taking something from the public repository, using their measurements, and now adding our measurement. Actually, it's using their measurements twice because they also did that, and just combining them together and learning something new, maybe, or starting to learn something new. More data sets is the open cell from the Hans Zuckerberg initiative, which is also talking about localizing an interaction of cells. Basically, what they did, very simply, I'm downing what they did a lot, but what they did, they find that they did some sort of auto encoding, like we know, and then use the latent vector as a representation of the organelle localization pattern. And then they did similarity on that. So their question was, we want to define measures for similarity between different localization patterns, and they used for that auto encoding. And based on the similarity in the latent spaces, now we can define networks of which proteins are related to one another in terms of their location within the cell. The third data set that I wanted to show you is Open Organelle, which was developed by Janelia, which is also a philanthropic entity of Howard Hughes Medical Institute. Oh, previously it was Hans Zuckerberg Initiative, Zuckerberg, the Facebook guy, right? And this is what you see here. I didn't show you any of this data in this course. I think this is electron microscopy data, EM data. And they basically trained networks to identify within EM data the different organelles. So it's very powerful because you can see in one image, you can actually see all the structures, and you get resolutions that you are not going to get with the light microscope. The problem is that you cannot do live imaging, you cannot do dynamics, but this is fantastic. And what you can see from all of these data sets, these are really major data sets, and a lot of effort, tons of money to create them, and they are public and they are amazing. And you can see a common theme. All of them are interested in the organization of the cell, of a single cell. So it just gives you some insight on how big a question is it, right? We want to just understanding the organization of the organelles within a cell. Look, HHMI, Hans Zuckerberg, Allen Institute, and right, all of these are putting all their efforts and all their money in all their funding in order to learn this basic question on how a cell is organized and how the organization of a cell is linked to the cell function. Just giving you some proportions on how big is this thing. And okay, I'm going to stop here. I think that a big question now is, the big challenge now is to come up computational thoughts and start thinking what can we do with this data? What tools can we build? What knowledge can we derive from that? What new questions can be asked? How can we integrate? Can we use the Allen Institute data to make predictions regarding organelle interactions, and then go into the EM data and see that the interactions are actually happening at the high resolutions? So I'm actually organizing a subgroup in the American Society of Cell Biology in December. It was just accepted last week, exactly about that. So exactly about, I'm bringing the people who generated these data sets and building these tools that I showed you today. And I asked them to talk about how they see, to prepare talks on how they see the future of using this data. Because now people generate this data, it's saying, okay, let's generate it. But now what we can actually do is that I have a few ideas, and in the lab we're trying to do something with it. But I think the field now needs to really think about it as a community and think how we can move forward the field. Okay, I didn't leave a lot of time, but and we can move to Hebrew, and אם יש למישהו פידבק על הקורס, אז עוד פעם, בבקשה, קודם כל תמלאו את הטפסים של המשוב הזה, ודבר שני, כי זה תמיד טוב שיש גם כתוב וזה, כאילו, דבר שני, אני רוצה לשמוע, שכחתי לשאול גם על ההרצאות אורח, מי הם היו טובות ומי לא, אז אם יש מישהו מההרצאות אורח, אני אשים לכם כאן שהייתה לא טובה, אתם חושבים, אז תגידו לי, כי אני חושב לקראת שנה הבאה, אני אפסיק אולי את ההקלטה, שלא תתביישו, אני לא הולך להגיד לזה, אז טוב, להקלטה אני אומר להתראות שנה הבאה וכולי, הפסקתי את ההקלטה.